My job today is to ask some questions about the microbiome and probiotics and prebiotics, which may be of interest as the field is moving forward, and connect some of the dots. And there are several things to think about when you start connecting dots. There's nutrition, which includes the type of diet, the supplements, and I will talk to you about supplements a bit. Prebiotics, the host environment, the types of probiotics. But in the microbiome, especially I think what is of some concern is how variable it is. And to get to a quantitative construct of the microbiome, which is to actually look at the actual biomass, and if you want to get into that kind of thing, you then begin to think about things like colonic transit time in the intestine, and where in the gut you're actually getting everything, the samples, and eventually for health. And for health, everything. So this is a problem actually. The constructs are many. And yesterday I noticed that there was always this tension between thinking about probiotics, for example, in the framework related to disease, or was it in the framework related to health and the reduction of risk as one aged, for example, or some even felt it could promote functionality and good health. So these constructs have to be clearly defined when one goes forward, and the dots are all over the place. So I thought I will first, because I'm the first talker on a lazy morning, is to tell you a little about history, because I think this, if you look at the people who started this journey, you will see that the wide-eyed, broad vision that they had, and my plea is that that is the vision we should have. There will be some physiology here. Now, I want to start with Burkitt, and I owe some of the slides I will use to John Cummings, who gave the Gopalan oration this year. As an old friend, he was somewhat a mentor for me when I was a medical student, and I'll tell you why. Now, when we were all medical students, Burkitt was, of course, the talk of the town. He was famous for having a cancer named after him, but he also started writing a lot at that time about the ills of a Western diet. And it was funny because he was a surgeon who went to Uganda and began to practice there, and noticed that the diseases he was seeing was very different from what he saw when he was practicing surgery in UK. So he actually wrote to his brother, who was still back in the UK, and told him, let's both collect a series of 300 cases of acute abdomen, and let's just note them down. It's not 100, it's 300. I made a mistake writing it there. And after 2 years, they collected the cases, he and his brother, and he found that almost 230 or 300 cases in the UK were acute appendicitis. Whereas in Uganda, there was almost no appendicitis. Indeed, the commonest for some reason was strangulated inguinal hernias. So he clearly said, look, there's a difference in what I see over there, and I trust my brother. And he wrote a paper on this in the East African Journal. And his conclusion was that the African Ugandan diet was an unrefined diet, whereas in England they had introduced the roller mills to extract flour from wheat, and it was a high extraction wheat, and it was very refined, and he felt there was something there. He didn't know what fiber was. He just thought there's something wrong with that diet, and that was very prescient of him. And he was, of course, a hero for all of us as we were studying pathology, where he really began to look at geographical pathology, and he figured out that the cancer that was named after him was occurring in certain geographic locations where there was a lot of arthropod-borne disease, there was malaria, and he started to look at that. And he was working with Sir Anthony Epstein, who, of course, was in England at the time, and he used to send Epstein his samples of lymph nodes. And the story, I won't tell you the story. You asked me to cut short, so I'll cut short that story. But it's a remarkable story of how the sample got delayed on a Uganda flight to the UK, and the cells began to separate and went into solution. And they were able to grow the virus in those separated cells. And his technician was Yvonne Barr, and that's why it's called the Epstein-Barr virus and the lymphoma. But the point is that Burkitt he was an FRS. He should have won a Nobel Prize, in my opinion, for this fantastic thing. The only person I think who eventually got a Nobel Prize for the infection and cancer link was Peyton Roux, who was working with chicken viruses. It's unfortunate that Burkitt didn't get it, but he was credible. He was really a credible person, and when this credible person begins to talk about fiber and something in the diet, everyone listened to him. He was an FRS after all. And so he then, at that point, went to a well, this is the cancer he actually became famous for, and this is his classic paper in which he spoke about his lymphoma, and it was a dramatic picture that he put up of a child with those two pictures taken 8 weeks apart, and to show how fast the cancer grew. Now, he then went to a gastroenterology conference in the UK, the British Society of Gastroenterology. By the way, this history is important, please, because it will lead to why I think we have to look at the microbiome a little differently. He went to a gastroenterology conference, and he met this man called Neil Painter. And Neil Painter was actually seeing a lot of diverticulosis in the UK. Diverticulosis is this disease in which the colon has little diverticuli popping out because of high pressures within. And what Neil Painter was doing was, if you can look at this, if you can look at the left-hand side, oh gosh. Okay, I'll use the pointer here. If you can you see this now? Yeah. If you can look at the top graph here, you'll see that Neil Painter had patients who had a bowel habit of, many days. It could be every 3 days or every 2 days. And these patients had diverticulosis and diverticulitis. And eventually, when he put them on a bran diet, you'll find that all these moved to the right, such that all his patients were passing stools once a day. And he actually sent this to the BMJ, published it. And the BMJ published it. And if you think about it, this was a trial which was uncontrolled. There was no control group. But his referee was Richard Doll. Now, I don't know if you've heard of Doll and Peto, the great epidemiologist of the UK who actually showed that smoking is injurious to health. But Richard Doll was the referee, and this is the greatness of Richard Doll. He said, the study has many faults, but I'm not going to throw it out because it makes sense. And I think that is what you like to see in a good referee, someone who just uses his own ideas. And he just said it was probably right, and he thought the BMJ should publish it. But the point is that Burkitt met Painter here and began to see the benefits of fiber through bran. And then, at that time, we were all thinking that, fiber, and if you went to houses and villages in India, you'd see cow dung put on the walls to dry, and then that cow dung was used as fuel. And you'd think, well, that cow dung contained a lot of fiber. And that fiber that you would put into a fire and burn. And that's how fiber worked. It bulked the stool by just holding water and just came out undigested. And then, because the MRC started getting very interested in what this FRS Dennis Burkitt was saying, that there's something in fiber, the MRC got John Cummings to begin to investigate this whole thing. And when he started investigating it, he looked at stool weights and he looked at transit time. And it's important for you to know that Dennis Burkitt, all his work included him going to villages around the capital, Kampala, and actually weighing stool samples. He used to collect stools, but not a small sample. He collected the whole damn stool. And he used to actually measure transit time. And those of you who have ever measured transit time will know it's not easy in the gut because it kind of mixes up as it goes. So a marker is very difficult to use. You need to use pharmacokinetics. And you actually create a steady state of markers in the gut, and then you can actually calculate clearance rates. I've done this, so I know how complex it is. But Burkitt actually did this, and it's remarkable that this man who discovered a cancer actually went around collecting stool samples. And you'll see here that these stool samples in the UK were, well, in Scotland and UK were a measly 100 grams per day. And as you came down to India, it was in Delhi, it was about 300 grams per day. And in Uganda, it was almost half a kilo per day. So it tells you that there's a huge difference in the amount of stools one produces. And you begin to think, gosh, if there's a 5-fold variation in stool weights, maybe the last thing I should be doing if I want to do inter-country comparisons is to take a small sample of that stool and measure the microbiome. I need much more quantitative determinants rather than a qualitative relative microbial prevalence. And I'll come to that in a little while. But the transit times in India was about, and I've measured this, is about a day, 24 hours to 36 hours. And they were finding that in the UK it was about 3 to 4 days. So there's lots of time for things to happen in the colon for the UK people, but not a lot of time in India. Now, the important thing was that John Cummings actually found, and this is a paper that got me very interested because it was a follow-on from Burkitt's work, and that's the point, that it actually discovered what he called biomass, or the microbiome in those days. And what he found was that, and if you look at these pictures here, you can feed people these non-cellulosic polysaccharides, or you can feed them cellulosic polysaccharides, and you'll find that if you feed them a lot, unlike what you expect to happen with cattle or cow dung, where you see a lot of fiber in the feces, in humans for sure there was much less. There was these shaded bars of what was excreted in the stool. So the stool weight did not contain what you fed. There was something happening to that fiber that was being given, or the polysaccharides, they were being degraded. And John Cummings wrote this very nice paper in Nature in 1980, which actually talked about the mechanism of action. And I think it was the first description of the microbiome existing and actually breaking down cellulosic polysaccharides and non-cellulosic polysaccharides. At the time, I was a medical student and I was very keen to do some research. And I went to the physiology professor, Dr. Shetty, and we decided that, well, this is what John Cummings was finding, we should also do similar things, and we actually wrote to him and we began a series of experiments. But the point in the dots so far is that it was found that fiber was an important substrate. Processed foods were bad, the bacteria were involved, it wasn't just a physical action, and if bacteria were involved, they needed time, so the transit time was very important, fecal bulk was very important, and there was a role of microbes. Now, the point was that if you say microbes exist in the colon, someone's feeding them. And what and how are they being fed? And let's get quantitative again. How much do they need to be fed? They all have nitrogen in them. Where do they get that from? And if you feed them and they metabolize that, what comes out of that feeding? These are important questions if you're going to connect the microbiome with health or even disease. Now, just go down to the bottom of this slide, for example, and this is how much, if you can see that. And in the UK, John Cummings actually figured out the biomass that was being excreted on that 100 grams of stool or so per day. And he figured that that was about 15 grams of bacteria were being excreted per day. In other words, that was the steady state of the microbiome. 15 grams went out, 15 grams reappeared. And that meant that you had to fuel that reappearance, and that meant you needed about 50 grams of biomass. And this is where I think it's really important, and I think in India, I hope this happens, that the ruminant physiologists and the ruminant people who are interested in ruminant metabolism begin to integrate with people doing microbiome work. Because the ruminant guys, and there's Dr. Prajapati somewhere here, and I think he'd be interested in these things. The ruminant guys were the ones who were actually working out what they call feed conversion, because that's what you want to figure out in cattle. How much do I feed? How much does it convert into? Eventually, how much milk does that become? And that conversion is what we need to figure out for ourselves, because for 15 grams per day, you need 50 grams. Even that 50 grams, I will put it to you, none of you eat more than 30 grams of fiber per day. So where on earth does that microbiome get that energy to actually grow? 30 grams, some of you probably eat only 20. This is the problem. Now at the time, there was a person in the Mayo Clinic, a gastroenterologist called Sidney Phillips. And Sidney Phillips, along with one of, I think you knew him, and he was very nice because he actually put in a long tube down the gut, past the duodenum, right up to the end of the ileum. And he then sucked a sample out of ileal fluid and measured the starch in that, not fiber, starch, 1,4-alpha linkage sugars. And he found that up to 20% of the starch that you eat can escape your amylase action in the jejunum. So there's a significant amount of starch, not fiber, that also gets into the colon. I wrote to Sidney Phillips and I asked him, can I have one of your tubes? And he very kindly sent me one. It was about 5 meters long. It was a long tube weighed down by a bag of mercury. And I just couldn't get approval. And I was scared because there was a bag of mercury tied up at the end, at the tip to pass down as a weight. And I was just scared, silly, that that bag would burst inside and you'd have mercury flooding into the intestine. But that's what he did. And I still have the tube and never had the courage to use it. But that's the point, that we thought, well, in India, we're getting 300 grams of stools per day. And we're eating not enough fiber. So is it possible that it's the starch that's working for us? And we did this experiment at the time with Prakash Shetty, and we published it in the American Journal of Clinical Nutrition, that we fed subjects with cornstarch, a terrible looking dish, which was just like, you know, you've seen cornstarch if you thicken it with water, it's like eating gum. But our subjects ate that gum, and you know, their stool weights went up. And it was a nice little study that actually proved that it wasn't just fiber. And if you look at us in India, we eat 400 grams of cereals per day. And of that 400 grams, even if, say, 10%, 40 grams of that cereal just gets passed and escapes digestion, that may be a good deal. But let me get back. And what I want to get back is that there's a component of sugars in your diet which is now becoming greatly important, and I think is a fascinating subject. And that is the oligosaccharides. So other than fiber, the oligosaccharides, and there are lots of them in pulses, and these have sugar linkages that the amylase, pancreatic amylase, cannot break down. So they go straight to the colon, and they are really good. And that's why we always tell people, eat pulses, it's good for you. But you know, the amount of these oligosaccharides in pulses is not great. So you have to eat a fair amount of pulses. And you don't want to eat too much of these. You don't want to go above 20 to 30 grams per day of oligosaccharides. But if you do, you will actually stimulate your microbiome in some good way. So I think these are important. And of course, the work of Dr. Ramakrishna, who has often worked with resistant starch, which you will find in raw bananas, or you'll find it to a small extent in potatoes or rice that you cook and cool. These are also very important. So I think resistant starch is one way to do things. Finally, if you don't feed enough of these, please remember that the bacteria then will turn on you. And when they turn on you, they're getting their food from the mucus. And this is called mucus grazing. And this mucus grazing actually reduces the kind of protection you would get. And it's not a good thing. So there's nothing like a healthy microbiome, but you've got to give it the right food. If you don't, you're in trouble. Well, I actually stopped doing this work. I felt like this girl over here running to collect stool samples, because that was my life when I was doing these experiments, running after my subjects with a bucket, making sure they didn't pass stools without telling me. Now, the whole thing about giving these different nutrients is that you can have one nutrient or an oligosaccharide like inulin, for example, stimulating one bug. And I think this is a precision that one needs to now follow. I think the days of us saying, well, I just gave more food or I gave more starch is one thing, but you really need to have a dose response of how much you gave to how much each bug grew. And these experiments are going on, and you'll see that soluble fiber, for example, can change specific strains. So you'll find that inulin, which is a polysaccharide, will come out and give you a change in one specific bacteroides, but cellulose will give you much less. By the way, it looks like cellulose is giving you a big response here, but the y-axis is actually just inflated. Now, where does nitrogen come from? You've given them the energy. Where does the nitrogen come from? Well, the bugs can get nitrogen from the protein that you eat. Now that assumes that you don't digest your protein very well, so some of that protein comes. But these bugs can actually, urea is a small molecule that diffuses happily back and forth between the body pool and the colonic pool. So these bugs have that urea, and they have the enzymes that can break down the urea and incorporate that nitrogen into their own body proteins. So that can happen, and many gastroenterologists, of course, use this when they feed fiber like lactulose in an attempt to trap nitrogen in patients who cannot do it in their liver. And here's the last thing, that when you actually feed these things, what comes out? Well, the important one that comes out is the short-chain fatty acids, and good luck to you if you want to measure those off a blood sample. You'll never see them. You'll probably only see acetate, because butyrate is cleared at the colonic mucosa. It's a substrate for the colonic mucosa to grow. It's actually anti-neoplastic in cellular cultures. Propionate is cleared by the liver, because it stimulates gluconeogenesis. But acetate is the one molecule that does pass the liver and is implicated in the gut-brain axis and has its own functions. But there are bad metabolites. Protein is not necessarily a good thing. We keep telling ourselves, oh, look, we don't eat enough methyl groups. One carbon metabolism is very important. We should eat more choline. Well, these proteins, choline, they can produce dangerous molecules from the microbiome. And one has to think about that when one thinks about the Western diets. And protein fermentation doesn't necessarily give you nice things. It gives you gases like H2S, an increase in certain types of bacteria, and it's not great. But the important thing with all this is that I was involved in deciding the protein requirements for human beings at the WHO, and I was the rapporteur of that group. And we agonized about the protein requirement of the newborn infant, because when we did it and we calculated it on a factorial basis, we got a value that was higher than what breast milk gave. And that worried us no end, because if the requirement was higher than what breast milk was providing, were we saying breast milk was inadequate? And we ran around this for a long time with George Beaton's analyses and so on. And eventually we realized that there was a component of nitrogen in the breast milk that was not protein, it was urea. And if you actually converted that urea into amino acids by bacteria working with the human milk oligosaccharides, then you could meet the protein requirement. And that's why we dodged that bullet, in my opinion, because we were really worried that we were calculating the requirement for infant protein to be higher than what breast milk gave. But when you calculate it as nitrogen, it works out. And I just want to end with this, and this is the work of Professor B.N. Tandon from Ames, who actually did this great work where he looked at the digestion of proteins and fats in undernourished people. And he actually gave subjects pancreazymin and secretin and actually intubated their duodenums and was able to aspirate the duodenal juice, pancreatic juice. And he found that if you were undernourished, you didn't digest things because you didn't secrete enough pancreatic enzymes. So I think that's an important thing in India. This experiment was repeated in Africa and in Paris, and everywhere it's shown that populations that are slightly poorer have much lower pancreatic function. It's called a silent pancreatic insufficiency. I think it hasn't been investigated in India because you won't get ethical approval to do these experiments. But nevertheless, it tells you this, that your gut and the colons of Indians particularly, I think is a slightly different place. The substrates reaching it are different, lots of cereals, poor digestion of protein. What's going on over there? The fecal metabolome is an important step forward from just looking at what's the metabolome. I think there are lots of dots over there. So at this point, I think what the science is really the bottom of the slide, which is what's the composition of the microbiome? To me, that's about 1/10 of the answer. What really is important is the whole business of what that means to colonic physiology and health. And that includes the colonic transit time, the substrates that get into the colon, the substrates that come out of fermentation and how they are absorbed and how they affect each other. And of course, don't forget the bulk of that stool. And there are, toilets now that, by the way, Google just filed a patent on a toilet that could measure your health. And presumably one of the things that would measure would be fecal weight. And the Japanese toilet, I believe there's somebody in Japan who's actually working on such a super toilet that'll probably tell you didn't eat enough fiber and because your stool weight wasn't good enough. Maybe, but that's apparently going to be there as well. So this is all the future, that there will be precision medicine linked to all these things. But the composition of the microbiome, in my opinion, is the start. It is nowhere near the end. Now, the key issue is if you're going to then just measure composition, I feel there's several problems there. Remember, we are not measuring what I think is colonic health. You're just measuring a microbiome, and you're taking a speck of stool and you're going and doing a relative microbial profiling. Not a great thing in my opinion, but good for you. You must do it. It's a good start. And the problem is that this is a relative microbiome profiling, and I was privileged with Dr. Rajiv Khosla and with Neerja to be in the Yakult conference this year, where this person, the last name author, Jeroen Rees from Belgium, has this Flemish cohort. And he's actually presented to me what I thought was the next big thing. And you may want to argue with me, but I think he did a great thing in that he actually measured the bacteria by flow cytometry. Beyond just measuring the DNA or the RNA, he measured and counted the bugs. And in doing so, he came up with a quantitative microbiome. I urge you to read this paper, which just appeared, I think, this year in Nature. And it's fantastic because it shows you this is relative microbiome profiling and this is quantitative microbiome profiling. And you will see that what you thought was a difference in bacteroides between Crohn's disease and controls may not be there. And on the other hand, if you saw differences in Prevotella in Crohn's disease by relative or if you didn't see these differences, you actually find, well, perhaps they are there. So there's a restructuring of this whole thing. And I think this is the kind of work I really, for me, I'm the guy who measures stool weight for a living. And for me, quantitation is critical. So that's very, very important if you're going to look at this. And of course, you just take a single stool sample or even just take a perineal swab. That's not good enough in my opinion. But even if you did that, you took one single sample. There's again the same group, I think, in the Flemish cohort, in which they've actually looked at the variability of this biome and find, to their surprise, that they can only explain about 16% of that variability based on the kind of phenotyping they could do. And what's the phenotyping? It's mainly medication, at least in that country. But of course, I think diet and lifestyle are equally important. I believe you're going to be doing that in the Human Microbiome Project. Good luck. I have a lot of questions to ask about that. So here's the point, that you really need to look at the variability between individuals, the variability within individuals, and I want to show you this study. I'm sure you've seen this, the study done in Guangdong province in China on 7,000 odd individuals, in which they looked at different districts within, or they called it areas, and you'll see that this coloring is socioeconomic status, and they found that variability of relative microbial prevalence, and I will use that word, it's relative, was really by district. And in those districts, it was next by occupation, or by education, or by age. So it's like, good luck again trying to characterize a human microbiome, because it's varying by district. And that you don't know what's happening within those districts. So to me, 7,000 subjects, that's an expensive study. To me, I would demand great rigor in the sampling of individuals, because the household seems to be the unit, and after the household, it's the village, and then the locality, and believe me, you've got an epidemiological nightmare on your hands, and we're still talking about relative microbial prevalence. I really wonder where we should get to if we want to take a step beyond, because I think India should do this, but I think it should do it in a different way, or not different way, it should do it right. All right, and of course you know that India, by the way, do you know migration in India is enormous? Do you know that in India, 50% of migration, and I think about 10% of our population migrates every 10 years. That's an enormous amount, okay? You're talking of 1 billion people, 10% migrating between censuses. 50% of that, by the way, is rural people migrating to rural places. Do you know why people migrate in rural places? It's because of marriage. And here's my point. Provinces show differences and migration shows differences. Migration of Hmong women and Karen women of Vietnam into America shows that they lost diversity within 6 months of reaching America. And I bet you that if you don't sample your people as unmigrated and migrated, you're going to have trouble, because that's going to be another problem. So you have to phenotype ever so carefully if you're going to do a human microbiome project in India. So that's about the migration. I'm going very fast, and I'm going to finish. I have just 2-3 more minutes. I just want to tell you that diet, of course, really alters this. And should you be, after eating Neerja's fantastic dinner last night, heavy meat, where I think my meat intake went up to 30% of my calories. Well, here's what happens when your meat intake increases 30% of your calories. It jumps up in the next 2 to 4 days. It's very rapid, but the good news is it comes down again. Here's the bad news. If you took my sample today or tomorrow, you get a different biome from what my, sorry? Will decrease, that's right. But the beta diversity increased, the alpha decreases. So between days, it actually increases. It's very counterintuitive. I couldn't understand this paper, but here is the meat experiment. And you'll see that the beta diversity that is between days actually increases. The alpha diversity is roughly the same. So I think your diversity is another question I want to ask. Again, this is relative microbial prevalence. You're looking at relative diversity. And so I'm not a microbiologist. I really think it needs expert help from you. But the point I'll make is that the gut microbiome can rapidly respond to diet. And this is potentially good for you if you want to sell someone a probiotic, but it's also bad for you if you want to characterize variability. And this is, of course, seasonality variations. I saw some poster outside, someone was talking about it. But this seasonality is important because in the wet season, your diet changes. In the dry season, hunter-gatherer type work, your diet changes again. And you can have that variability. So there's another problem. I just want to end by telling you that there's some untranslatable stuff, but the most interesting thing is happening with the microbiome. It's a dot that I would love to investigate, but you can't do it in humans. And that is the kind of work that goes with longevity and the whole business of increasing the longevity of fruit flies, for example. That's one thing that actually looked at fruit flies along with some Ayush products, which actually increase their longevity. You can see it here. And this one where they lived for 70 days as opposed to 40 days in the other one. But there's another interesting study in these little fish, in which fish who were given fecal transplants effectively of younger fish live longer. Now, does that mean that you have to chase young fecal transplants? I don't know. But eventually you'll never prove these things because you can't. But they raise interesting problems. And of course, we have to chase them down with short-lived animal models, but eventually we still have several health dots to connect. I want to ask these questions. First, get your frame correct. Are you talking about health, or are you talking about disease? If you're talking about curing a disease, it's a different frame. If you're talking about preventing disease, a different frame. And as Dr. Selvamoorthy asked, if you're promoting function, it's a different frame. Get your frames correct, then I have no problem. Is there a core or a mean microbiome? Are we chasing this for, is it just a kind of a fantasy? I don't know. The core microbiome, in the first place to me, is too variable. And unless you characterize your subjects, it's very hard to know. So should we look for it or should we embrace that variability? Can we seed the microbiome in a stable way with appropriate probiotics? Well, we know, yes, you can seed it, but it eventually reverts to what it was before. And one asks the question, is it the virome that is what is actually creating stability of the bacterium? And that's another question which I think you should answer for me. What feeds the microbiome? If you're going to get into this probiotic business and prebiotics, We need to get the amounts right. I'm a quantitative physiologist and a quantitative nutritionist, and it makes no sense for me to say eat fiber. Next question, how much? Which fiber? How much? How much? How much? All the time. And the time course of how you give these things. And food fortification. India is fortifying its foods. There's so much iron in the system. There's a paper coming out in Journal of Nutrition, which I wrote, which actually talks of the risk of us exceeding the tolerable upper limit of iron intakes with one fortified food like salt and the NIPI program, which provides iron supplements. And we give that iron. 50% of women in Rajasthan are at risk of exceeding the TUL. That's how bad it can be. That, when given to a pregnant woman, will change her microbiome. That microbiome will transfer to the child. Yesterday we heard so much about how the woman's microbiome gets into the child. Well, it's an altered biome. It's an iron-based biome. And what that does to future generations, I have no idea. But food fortification is something we should not take lightly and be careful, particularly with pregnancy. There's a very nice article that came out in Science just recently. You must have seen this. It's a review on gut microbiota at the intersection of diet and human health. I enjoyed reading it. And of course, there's great excitement about all these things. But just like me, that article felt that there were fundamental questions regarding the microbiota and its relation to one side the diet and the other side to health. And it's really difficult to get your fingers onto that. And they talked of this thing called the Anna Karenina principle. The Anna Karenina is a Leo Tolstoy story in which it is said that all happy families look alike. Each unhappy family is unhappy in its own way. It's diverse. And they apply this now, this Anna Karenina principle, to the microbiome. And they say that dysbiotic individuals vary more, not diverse less. They vary more in microbial community composition than the healthy individuals. So I would ask you, diversity, I think it's a very, very difficult question to answer, but let's think of Anna Karenina as we go forward. Thank you. Thank you.